All right. Thank you all so much for attending. Thank you for people who are online. It's really great to be here. As Josh mentioned, my name is Emma Rosi. I've been a scientist here at the Cary Institute for 14 years. Before that, I was at Loyola University Chicago as a faculty member. And I'm going to kind of give you a little bit of a rundown of what I've been doing all these years quickly. And I do want to point out that there are a number of paintings in this seminar that are mine. So yeah, we'll just keep going. Okay, so I don't know why I said I would do this, give this seminar, but because no one else has done this, but somehow, like no one else at the Cary Institute, but I thought, well, I should come and summarize my career. So then I was like, well, how do I do that? What do I say? And so I took the traditional approach in the beginning and thought, well, okay, so first of all, I got to summarize that I've worked with so many great people. I need to thank all the funders, my many collaborators, which I could not possibly list here, and co-authors, but I do want to give special thanks to the graduate students, postdocs, research support specialists who made it so getting up in the morning was worthwhile. Like they're just amazing, all the people I worked with, and you're going to see some of their work highlighted. Unfortunately, I can't highlight everyone's or else we'd be here for today and into tomorrow. So I can't talk about all the work that everyone's done. I'm just going to focus on some highlights, but I just do want to thank all of them. This wouldn't have been possible without them. The other thing I did is like sort of a standard, well, let's see, what do I do? What is my research actually, what is it? And it turns out when I say I'm a river and stream ecologist, I am right. I really am a river and stream ecologist because I took all the titles of all of my publications, I made a word cloud, and you can see that like the biggest words that appear most are river and stream and ecosystem. So I'm a river, stream, ecosystem ecologist, and then a whole bunch of other things that I've done work on. So this just kind of gives you a sense of the diversity of topics. And then I did this, which is I took all of my publications and I said, okay, well, how about by habitat? And, you know, I've done work in urban systems, large rivers, forested streams, agriculture, a bunch of papers that are just ideas or synthesis and methods. When I think about publications by topic, you know, I know that a lot of people know me from my work on contaminants, but it turns out I've done an equal number of things on nutrient cycling and food webs and all these other things. So, you know, but this doesn't really make for the most exciting, interesting talk. So then I was thinking, okay, well, really, like, what am I going to talk about? You know, how am I going to fill this time? And so I have, if you forgive me, I have changed the title of my talk a little bit to say, what the flux have I been doing all these years? And I'm going to explain to you what I mean by flux. The thing that gets me really excited in science is when we can actually estimate flux. And I'll define that in a minute. As an ecosystem ecologist, I love to think about the movement of materials. And so I'm going to focus on this seminar. You know, I could talk about all different things from my research portfolio, but I'm going to focus today on the research that I've done that uses flux, and I'll explain that, and kind of highlight that part of my career because it's been very exciting to me. So if you just look at the definition, there's a whole bunch of definitions of flux. When ecosystem ecologists use this term, what we mean is the rate of transfer of a fluid or particles, material or energy through ecosystems, from one ecosystem to another, that kind of thing. So this last definition here. And so that is what I mean by flux. And why do I work on this? This has been sort of a cornerstone of ecosystem science from lichens, gene odom. So I did my PhD at University of Georgia in what is now called the Odom School. So I learned a lot from odom and many people, and of course, gene lichens. And so just to give an example, the Hubbard Brook Experimental Forest, we have been measuring, Gene Likens and his colleagues in 1963 came up with this idea, like, well, we can learn a lot about the ecosystem. If we measure the inputs and the outputs, we can learn about what's going on in the watershed. So by looking at the flux of material in and the flux of material out, we can actually learn. The study is not just about budgeting. It's actually a way to learn something about the ecosystem. And so I've been lucky enough to be a collaborator on this project. And this year we celebrated 60 years of sampling every week, never missed a week. It's kind of amazing. And so you can learn about that if you're interested. I'm not going to talk about that work today. But When I took ecology class as an undergrad and I learned about the Hubbard Brook experimental forest, I wrote in my notes, this is the kind of thing I want to do. And that, I mean, and I work on this project now. It's kind of amazing. So I've gotten to do this kind of work, and it's really inspired me throughout my entire career. So I'm going to talk about this. And here I am, almost 30 years ago when I started graduate school. I dug through my old photo albums and found a picture of me. So when I started in science as a graduate student, I studied the flux of organic matter through food webs. So the organic matter is a proxy for energy. And this is from my first publication that I wrote with Bruce Wallace. And this is a very busy diagram. I'm not going to go through it. But what we did was we measured how organic matter moved through food webs along a river continuum. So these are 4 different food webs, and you can learn a lot from doing this. And so I think this has like gotten into my brain as the way that I like to do science. And a lot of my research projects don't involve measuring flux, but the things that really, where I get really excited and animated is when I get to do this. And it's really important to recognize that it's not just about quantifying fluxes, it's about what you can learn from quantifying fluxes, what you can learn about from doing this that really gets me the most excited in science. So I'm going to talk today about some things that we've done to do this. So I'm going to give 3 examples, what we can learn from measuring fluxes. I started with like 9 examples, and then I had to pare it down. So I apologize. I'm just going to focus on 3. This is work that we did on wildlife in a river in Kenya, work that we did in the Colorado River that was focused on an endangered species, and then work that we did on urban streams in Baltimore and in Australia, where the platypus lives. So I'm going to give you some examples of what we can learn from measuring fluxes. And I want to stress that it's not just about, the budgeting and it's not the fluxes that we care about, it's what we can learn. It's a pathway to discover new things. So the first I'm going to talk about is how fluxes can give you insights into the control on ecosystem patterns and processes. So that's sort of a first example I'm going to give you. And then I'm going to go through, and these are the 3 that I'm going to talk about, how fluxes can provide insights for conservation and management, and how they can tell us about threats to ecosystem health. Okay, so the first project I want to talk to you about is a collaboration that I did with Amanda Sobolewski, Chris Dutton, David Post, and others in the Mara River in Kenya. And basically, we were trying to understand, and what we were focused on was, the hippos and wildebeest live in this river. I'll tell you a little bit about that. What we wanted to do was quantify material fluxes, like how much are these organisms contributing to the river? And then importantly, what is the role of them in influencing ecosystem processes, how the river actually worked? And so I want to thank the funding and all of these collaborators that worked on this project. And I'm going to just share some insights about what we did. So first, we studied hippos. Not first, we did this all at the same time, but I want to tell you about hippos. This is the story I'm going to tell you the most about. So hippos in the Mara Basin, those are not rocks. Those are all, hippos. They wallow during the day in the river because they get really hot. If they're out in the sun, they actually can get sunburn. And so they spend all day in the river and during the day. And at night, they go up these trails, they carve these trails, and they feed on grass. And I'll talk a little bit about that. In the Mara Basin, it's home to about 4,200 hippos or so. There's various numbers, estimates, but about that. And this number has increased from the 1950s. In the 1950s, there was an anthrax outbreak, and there were only 100, 200 hippos, and now there are many, many more. And this is a map showing all the different hippo pools along the Mara River, so you can see all these hippo pools. This is work that was led by Amanda Sobolewski. She was a graduate student at Yale at the time, and then she joined my lab as a postdoc, and now she's at University of Florida as a faculty member there. And so what we wanted to do in this is we wanted to, the flux we were trying to quantify is how much do the hippos deliver of nutrients, carbon, nitrogen, phosphorus? How much are they adding to the river? And the reason they're adding this to the river is because at night they graze on the grass, and during the day they come into the river, and hippos have this weird thing that they do where when they poop, they like swish their tail around, they stick their butt out of the river, and this is a hippo in mid defecation, I guess. And so here they are, there's a lot of them, and that is a lot of material. So they spend half their day in the river, and so it's a lot of material. So Amanda set out, and all of us together set out to, like, how are we going to measure how much the hippos are adding of carbon, nitrogen, and phosphorus? And for those of you familiar with this, aquatic ecologists have tried to figure out, like, well, how much fish poop gets into this system? So they take a fish and they put it in a bag, and then they like wait a while, and then they measure the nutrient concentrations after an hour or something. But it's really hard to put a hippo in a bag. You know, they don't like it. They're really aggressive animals. And so Amanda had this brilliant idea to go to the Milwaukee Zoo, where there were 2 hippos that were in captivity. And basically every morning they clean the hippo pool, and it looks like this. The hippos go, they spend all day urinating and defecating. And at the end of the day, they go back through this cage, and Amanda and Chris went there with shovels and wheelbarrows and figured out just how much they added. This one day of hippos pooping in the river, in this pool. But with that, we were able to actually estimate this, which is a hard number to get. It's a hard number to get. So we figured out that, so based on literature surveys and things, each hippo in the Mara eats about 3 tons of grass each year. And the combined contribution of hippos, given how much time they spend in the river using game cameras and the data that we got from the zoo, they contribute about 13,000 tons of feces, 1,300 tons of carbon, 200 tons of nitrogen, 20 tons of phosphorus to the Mara River. That's a lot of material. So that's our flux. We've now estimated that, which was hard to do. But then that we don't stop there. We want to know, what is the effect, right? So like, yeah, that's interesting. It's a cool paper that we were able to estimate that. But what is the effect of all of this hippo activity into the Mara River? And this is a paper that was led by Chris Dutton, who also is a PhD student at Yale University. And he published this paper about organic matter from hippos causing a subsidy overload. We call this subsidy overload, and I'll tell you a little bit about that. So here's Chris Dutton. He's now a faculty member at University of Florida, and he was here at the Cary, and I was on his committee at Yale. And basically, he put dissolved oxygen sensors into the Mara River because we wanted to measure the metabolism of the river. So people might be familiar with this. You just put a dissolved oxygen sensor, you measure the dissolved oxygen over time in a river, and you can estimate the primary production and respiration. But what Chris observed is the following. Sometimes, so here's the river, oh, okay, oxygen going along like a normal river, and then sometimes it crashed. And when we say crashed, I mean, it's like it went down really low, very little oxygen in the river. And in fact, we observed, which is a hard thing to observe when there's lots of wildlife around, but we did observe fish kills actually coinciding with those crashes of dissolved oxygen. And so we found that these flushing flows, the dissolved oxygen created a stress to fish. And so then the question is, why is this happening in the Mara River, and are the hippos possibly to blame? And so we know they add a lot of carbon and nitrogen. Are they the cause of these DO crashes? And Chris, we all set out to answer this. So we had to answer some questions. Well, what's it like in a hippo pool? I'm going to explain why that's just a hard thing to know. And then could the flushing of the hippo pools cause enough of a dissolved oxygen sag to result in this big river? I mean, it's a big river. Can that actually cause this low oxygen? So let me show you a little bit about what it's like in a hippo pool. Here's a hippo pool that we worked in. And see, here are the hippos. And we're over here on the bank. Hippos are scary enough. But if you zoom in, you'll see that not only are there hippos in the hippo pool, there are now crocodiles that are about the size of a canoe that are also in the hippo pools. So we normally, as a stream river ecologist, I put my waders on, I go in, I take my sensors, I collect some samples. You can't really do that in the Mara, in these hippo pools, because dangerous wildlife there. And so what we did is we actually had this built that was this robotic boat. It was like a remote control boat that had all these sensors on it. It was built by this company Platypus in Pennsylvania, and they built this boat for us. And so Chris did all these surveys of hippo pools. So here's a pool without hippos. You can see the depth profile that we get from all these sensors, and then the conductivity. So this gives you a sense of how much stuff is in the water. I'll just leave it at that. And a pool with a lot of hippos, you basically get chemical stratification. So lots of material, low dissolved oxygen concentrations as well, and lots of accumulated products of microbial metabolism are in the bottom of those hippo pools. Because the hippos, so this is hard to know what this is. What is this boat going through? That's that same hippo pool. The hippos are over here. We drive this boat through. That is all hippo poop. All that stuff floating on the top of the water. There is a lot of ammonium, hydrogen sulfide, methane, carbon dioxide in the bottom of a hippo pool, it turns out, when there's all those. So it's not just the hippos and the microbes together contributing and creating these conditions. And then Chris did some other cool things. We built this artificial stream facility in Kenya based on our artificial stream facility here. We call it our safari streams. We set it up under a tent. Chris made this all happen in Kenya. And what he did was he added both hippo feces, which you can collect on the sides of trees in the morning after they've walked around. And then he added hippo pool water. So the water from the bottom of a hippo pool, and that's here. He's added that here, and you can see it's turning this stream black. In a moment, the whole stream will be black. And if you look at the dissolved oxygen, and then over time, basically when you add that hippo pool water, you can get a dramatic crash in oxygen. Same with just the hippo feces, but it's more dramatic because of all those products of microbial metabolism happening. And then we did some modeling, some math, and basically we figured out that when hippo pool water, so when there is a rainstorm in the Mara Basin, when the hippo pool waters, when they get flushed into the river, when it exceeds about 11% of the Mara River discharge, you can actually get a dissolved oxygen crash. So check. It looks like it's the hippos that are doing this, creating these fish kills. So this massive flux that we were able to measure, then by doing these mechanistic experiments, and we did a whole series of experiments that are published in that paper, not just this one, demonstrated that these hippos are the reason and they're driving this ecosystem. I don't have time to talk about all the work that we did on wildebeest as well. Here's again Amanda Sobolewski. In the Mara River, you've probably seen it on National Geographic. The wildebeest migrate through the Serengeti. They cross the Mara River. You've seen these dramatic videos of them, all these animals crossing the river, crocodiles eating them, whatever. And basically what we did in this paper is we quantified, again, this flux of material by wildebeest carcasses. So here, this is a major drowning event. Those are all wildebeest carcasses in the river. This is a really big river. Those are all carcasses, and then there's vultures. And we did all this work that Amanda led on figuring out, exactly how much wildebeest enters the river and where does it all go. And so that's published in that paper. And importantly, what we found, and this is published in a number of other papers that Amanda has published, is that the flux of carcasses into the river drives productivity. So, and in particular, it's the bones. So if you pull bones out of the river, you'll find lots more algae growing on the bones than on the rocks nearby. The bones, Amanda did work to measure the decomposition rates of bones versus tissues versus muscles, which is not great. It's not fun field work, but she did all this, quantifying how long does it take for a wildebeest carcass to decompose. And the bones can last for many, many years. And in fact, if you go into the Mara River, it's kind of like a boneyard. There's bones all over the place from these wildebeest that have drowned in the past. And they're an important source of phosphorus to the river. So all of this research that we did to understand the flux of animal materials into the river, what we have found is that this flux of material drives the fundamental, the way this ecosystem functions. That is like a key insight that we learned from this work. And although it feels like, may seem like the Mara is far away and it's really different, there's wildebeest there, and what does that tell us about rivers in general? Well, it used to be in North America that we had these. We had bison in North America, many, many more bison than there are wildebeest in Africa today. There were many bison that we have basically nearly eliminated. And so they probably, prior to us getting rid of them, hunting them, prior to that, they probably would have been a major driver of ecosystem processes in rivers in the past. We know that they would have affected grasslands, but I think what our research shows from the Mara is that they would have crossed rivers, they would have drowned, their carcasses would have been a major source of phosphorus and carbon and nitrogen to rivers. And so that's an important role of animals that we've really kind of lost sight of because they were gone before river ecologists started doing this kind of work. Okay. Now I'm going to talk to you about, we're going to shift gears here, and I'm going to talk about how flux can tell us about, provide insights into conservation and management of species. So this work took place in the Colorado River in the Grand Canyon, and our focal species in this system was the humpback chub, which is a federally endangered species, pictured here. It's a beautiful fish. And basically in the Colorado River, so upstream of this image, which I painted, upstream of there is this dam, Glen Canyon Dam. It was the last large dam that was built in the United States. It was put in 1963. When the dam went in, the river changed fundamentally because of the big, huge reservoir upstream. So there was a fundamental change in the river. The river is now colder and clearer than it ever was in the past. Because it's a hypolimnetic release dam. It releases water from the bottom of the reservoir. The temperature stratifies. So the river used to go from 2 degrees Celsius to 27 degrees Celsius on an annual basis. That's a big change. Now it's pretty much always 9, 10, 11 degrees Celsius. So the temperature is really consistent now. That is very hard on organisms like invertebrates, right? So it's very challenging to have such a dramatic change. The river is also, most of the sediment that would have come into the Colorado River. So it was said, I think Mark Twain said, it's too thick to drink, too thin to plow about the Colorado River. It used to be super muddy, like the river used to be muddy, but you build a big, huge reservoir upstream, and 90% of the sediment that normally would have come into the Grand Canyon is now sitting at the top of the reservoir. So 90% reduction in sediment. And then the discharge fluctuations have been strongly altered. So it used to be when the snow melted up in the Rockies, you'd get these big, huge floods in the Colorado River, and then in the summer it would go low. Now, because it's a dam operating for hydropower generation, the river discharge fluctuates on a daily basis like this, and we are lost the really high highs, and in some cases we've lost the low lows. A result of this is that the communities now lack native species. So half of the fish species, there were 8 in the Colorado River before the dam went in, half are now no longer present in the river. So 4 remain. And so 4 out of 8 native species were extirpated. And then we have introduced a whole bunch of non-native species of fish. The invertebrate community, I don't have time to go into it, but they also have fundamentally changed. And I'm going to talk about some invertebrates later, and you'll realize that they are definitely not native to the Colorado River. It's really changed the river ecosystem. Just to give you a sense of what it was like to work in the Colorado River, here's Lake Powell. This was our study site. So it's 225 miles of river. That's a pretty long study site. We had to raft for 2.5 weeks down in the Colorado River. And to get to our sites, we sampled this project. We sampled for 3 years. Every quarter, we went down the Colorado River, and then at the top and the bottom sites, we sampled every month. Okay, it's a beautiful place to work. My heart is in the Colorado River in some ways. I loved it there. Love working there. I want to tell you a little bit about what it looks like. So below the dam, you may have seen this is a famous photograph of Horseshoe Bend. This is right below the dam. This is what the river looks like a lot of the year, almost all the time below the dam, because there's no sediment coming out of the dam. It's clear. This is weird. The Colorado River should not look like this. It's really clear. But then as tributaries come in, as you go further and further downstream, tributaries contribute sediment to the river. Although it's much less sediment than the past, the tributaries still deliver sediment. And so when you go even further downstream, the river is then turbid and murky in some parts of the year when there's rain coming in. Okay, so I don't have time to talk about all the research that we did in the Colorado River. There's no way I could talk about that, but I do want to say, I give a shout out to Wyatt Cross, who's now a faculty member at Montana State University. He was our postdoc who led this work. But what I'm going to show you right now represented a ton of work, lots of people, a bunch of co-PIs that are listed here, 3 student theses, and thousands of samples. And I'm going to boil it down to 2 slides. So forgive me for that. But basically, what we did is we measured the flux. We were measuring fluxes. We used the same approach in the Colorado River. And we calculated all food web fluxes for 3 years at 6 sites, which if you know about how hard this is really, really challenging. But we did it and we measured the biomass. This is the annual biomass of aquatic invertebrates. And then we measured the secondary production of aquatic insects and invertebrates in the Colorado River. And I just want to point out that the tailwater, so look, the patterns here with biomass and secondary production, which is grams ash-free dry mass per meter squared per year. So this is how much is being produced in the river. This is important. This is an important number. It took a lot of effort to get to this. That the secondary production is really high right below the dam, and those clear algae, there's a lot of algae there. It's fueling that food web. This food web here has got lots of biomass and lots of secondary production. Much of that is dominated by the New Zealand mud snail, which is clearly not from the Colorado River. So it's from New Zealand. The New Zealand mud snail dominates the secondary production right below the dam, and they drop off as you go downstream. And then this shows you who comprises all that secondary production, all these different aquatic insects, which I know you don't know all those names are, but I want to focus on Potamopyrgus. That's the New Zealand mud snails. So you can see that who contributes to the biomass and the production along the Colorado River changed as you go downstream. That's an important thing to take home. Because we measured the entire food web. So we did the fish production, we did the bug production, we did all the gut contents of the fish, we did all the gut contents of the aquatic insects. We got like the whole story. We were able to measure everything. And I recommend you look at this paper. There's a lot of information. I'm going to just cut to the chase on one take home from this paper, is that the fish demand relative to the prey production. So what that means is how much do the fish need to eat in order to be that many fish in the river? In order for that, so you take all these bioenergetics models. I can't really explain it all today, but basically we were able to calculate the fish demand, that's here, shown here in this kind of gray, versus the production of the prey. So that basically means the fish are eating like 16% of the production. There's a lot of bugs out there, right? Fish eat like 16%. In the areas below the dam, which is only basically rainbow trout. That's where this rainbow trout is. It's a blue ribbon fishery. So they're okay. They have a lot of food. The New Zealand mud snail, notwithstanding, trout, New Zealand mud snail has this really interesting life history. They can close their operculum. So that means like the little hole in the snail, they have this little thing that can close. So when they get eaten by a trout, they just go on like a joyride through their intestinal tract, and they come out that bottom end totally unscathed. So they live through. So like a trout might eat a New Zealand mud snail, but they get no energy from it. So it just goes right on through, and then the New Zealand mud snail comes out and continues to reproduce and live its life. But downstream, the story is really different. Basically, we've got the 4 native and then the 4 non-native. So we've got a much more diverse assemblage of fish. This is where our endangered humpback chub is. That's illustrated here. What you can see here is that the fish demand overlaps with the prey production, like entirely. Okay, so what that means is, and this is I'm just going to get one take home from this very large paper and all this study. A major take home is that where the habitat is suitable for the endangered species, including this endangered humpback chub, basically the fish community is totally limited by food, right? You can't so if you want more endangered species, you gotta have more prey. right? Like you can't get any more fish. They're eating everything that's available, because you have to consider like that, like an endangered species lives within a community of a bunch of other species, right? And so, you know, in order to get a higher population of this endangered species, we got to make more food, basically. And so since this paper was published in 2013, I'm happy to say that the US Geological Survey, led by my colleague Ted Kennedy, has followed up on all this. And basically the work that they've done in the last decade is trying to figure out, how do we get more food? How do we increase the base of the food web? And they've done all these really interesting experiments, research to try and figure out ways to basically alleviate this food limitation. How do we get more bugs in the river? And so you could follow up, and I can tell you about their research, but I haven't been involved in that. But another thing, because we had this super well-studied food web, we were able to do something else that is pretty cool that I wanted to talk to you about. And this is work that I've done in collaboration with my colleague, David Walters. We published this paper in 2020. You can basically, because we have the food web fluxes, we went out, and at the time, David Walters worked for the US EPA. He got a small grant to come down on the river and measure mercury and selenium in all the compartments of the food web. So he and I took all these samples, and we basically, because we had, or we actually knew we were going to have, at that time, we didn't actually, it wasn't all done, but we knew we were going to do it. So because we were going to have a detailed understanding of all the food web fluxes in terms of grams per meter squared of ash-free dry mass, so material fluxes, by measuring the mercury concentration in the bugs, in the algae, in the fish, we were able to actually couple those, multiply it through, and figure out how much mercury is moving through the food web, which is pretty cool. People haven't done this before. We also, I didn't talk about this, it's covered in a number of papers, including that one that I just showed you from Cross et al., that we took advantage of the fact that there was an experimental flood that happened smack in the middle of our study. And so we were able to, in this paper, look at how an experimental flood, which really fundamentally changed the ecology of the system, how did that change? How did the ecological changes influence how mercury moved through the food web? Okay, so we were able to take advantage of that. These graphs are really rich, complicated. There's a lot of information on here. So I'm breaking it down a little bit. Here's the tailwaters. This is just below the dam. Here's rainbow trout. And this is mercury flux through the food web. So before it was grams per meter squared per year of ash-free dry mass or material flux. Now, by some calculations and modeling, we basically figured out how much mercury is moving through the food web. And what you can see is a lot of mercury goes into this New Zealand mud snail that's illustrated here. But because so basically very little New Zealand mud snails are incorporated by the trout, because of that, remember what I just told you about the life history strategy of New Zealand mud snails, they're like a mercury dead end, right? So they're like a trophic dead end. Mercury gets into New Zealand mud snails, it doesn't really go up into the food web. Downstream, where we have this much more complicated food web, all these fish that I told you just a second ago are eating all of the prey, all the mercury that gets into this food web gets up into the trout, up into the fish. And we did this both, this is pre-flood, this is post-flood, this is pre-flood, post-flood. We did this for the whole system. What you can see is that with downstream of the dam, the trout, actually, their populations increased a lot downstream of the dam right after the flood. And New Zealand mud snails got exported and moved downstream. And so what we ended up with was, as a result of the flood, we got much more mercury going into the higher trophic levels. Downstream, basically, any bit of mercury that gets into the food web gets into the fish. And there wasn't really a noticeable effect of the flood on this. And this is just sort of our, I don't have time to go through all the graphs and analyses that we did for this paper, but I just want to show you kind of illustrating this point, which is that mercury flux was driven by ecological interactions, which as an ecologist that studies contaminants, this is very exciting to me because it says that the ecology really matters. It's the ecology which drives how contaminants move. And this is a really different way of thinking about contaminants. So below the dam, basically where there's just rainbow trout, as I said, the New Zealand mud snail is sort of a trophic dead end. The mercury just kind of cycles through those guys. And then all of these black flies, they just basically get eaten by the fish, and there's a whole bunch of mercury that goes to the fish. A little downstream, we did some really interesting calculations and figured out that a lot of the black flies down at that site, because the black fly production is high, but the trout are low during this time of this study. Actually, a lot of the mercury goes out of the river. These escape predation and they emerge and they carry mercury into the riparian zone. Well, why does this matter? Well, I didn't tell you there's another endangered species that lives in the Colorado River in the Grand Canyon. It's called the endangered willow flycatcher. Guess what it eats? Riparian emerged aquatic insects, flies. So we really have to be concerned about, are these potentially being exposed to mercury driven by this ecological interaction? And the reason that there are black flies emerging is because there's not very many fish eating this production. And as you go even further downstream, as I told you, all the mercury that gets into the food web gets into higher trophic levels. Okay, now my last example that I want to talk to you about, and if you're not overwhelmed with how much information, I'm surprised. I'm sharing a lot today. I want to talk to you about how fluxes can give us insights into the health of ecosystems and even human health. First, I want to talk about synthetic chemicals. This is a paper I wrote with my colleagues Emily Bernhardt and Mark Gessner, which, this is work that we pulled together and we wrote this paper that said, we investigated synthetic chemicals and put them up against other recognized agents of global change. And what we said in this paper is that synthetic chemicals are also agents of global change. They're also increasing exponentially, like nitrogen fertilizer, world population, CO2, all those things. So that paper is available if you're interested. What has really driven a lot of my research throughout my entire career is what are the consequences for the health of ecosystems when we have these synthetic chemicals getting into the environment. And so I don't have time to talk to you about all this work, but I had all these students and graduate students and postdocs, and then I had high school students and undergrads work in the artificial stream facility here at the Cary Institute, where we looked to see the effects of drugs on a whole bunch of ecological processes. And we found all these different things, like drugs affect microbial community composition, antibiotic resistance, they change the life history, they influence crayfish behavior. And you've made it in science when someone makes a cartoon out of your research. So this is a paper that was led by AJ Reisinger and Lindsay Reisinger, and this is a cartoon from our research, which is, I'll give you a moment to read it. So anyhow. Well, we studied the effects of antidepressants on crayfish behavior, and that is actually what we found. So it's really kind of cool to have a cartoon. I want to focus on the flux. So we also, not only do drugs get into water bodies and cause effects as dissolved concentrations, but working with Erin Richmond, who spent a lot of time here at the Cary Institute, I co-advised her for her PhD. She now works at the Australia EPA. What she did, and what we published in this paper, what we figured out is that not only do drugs, are they in rivers, they're getting up into the food web, and aquatic insects are taking them in. And so I'll just cut to the chase and show you the data. We looked for 97 drugs. We found 68 different drugs in aquatic insect tissues. So this is in hydrocyclids, and they accumulate in aquatic insect tissues 3 orders of magnitude higher than what's in the water. So what people often say is, oh, drugs, they're in such low concentration, nanograms per liter, is it going to have an effect? Well, the last slide, I should have said, we studied everything at nanograms per liter. So yes, it does have an effect. But not only that, once it gets into the food web, then the effects might even be greater because the concentrations are so much higher. We also, Erin went and collected spiders along the streams. I should say this was work that was done in Australia. So we collected these samples in Australia. Erin collected spiders and that eat stream insects, and they also have pharmaceuticals. So we found the same suite of drugs in the spiders. So they must be, the insects are taking these drugs, and it's a flux out of the river into the riparian zone, potentially affecting animals that consume them, like spiders. Using some understanding of metabolic, the metabolism of platypus and trout, we were able to calculate the flux, like how much drugs does a platypus get from eating aquatic insects. Why did we study platypus? I told you this was all done in Australia. In the streams we work, there are platypus. Platypus, you probably haven't thought about them at all. They're an egg-laying monotreme. They're really cool. They have little pouches, cheek pouches. They use that duckbill to scrape bugs off of rocks in streams. That's what they use it for, and that's all they eat are aquatic insects. And so we were able to take some literature values and some understanding of the metabolism of them, and then couple that with our measures of the concentrations of pharmaceuticals in the tissues of bugs to then do this. And that what we found, this is both for trout and for platypus. Platypus, just by eating aquatic insects that we found in the streams and measuring the concentrations of drugs, we found that, you know, if they're living in that stream, they would get nearly 60% of a human dose of antidepressants. What does that mean to a platypus? I don't know. Someone needs to figure that out, but I don't think it's a great thing. You know, like, I mean, they're definitely being exposed, and the only way we could get this is by measuring flux, right? And by doing these kind of this models. The other thing that is a real unknown, which is for the next generation of scientists to figure out, is what are the effects of exposure to multiple drugs? 69 different drugs. I mean, if you went to the doctor and they said, what other drugs are you on? And you'd say, well, I need some more pages because I'm 69. You know, they would say, that's probably not a good idea. We know that they interact. We don't really understand what happens in the environment. And now I want to highlight a paper that just came out a couple months ago that I did with Barbara Hahn and Jurker Fick, where we took this same thinking about flux. And Barbara had been writing papers about, you know, these amazing papers that I read of hers, because she's my colleague here at the Cary, about the importance of bats and how bats you know, harbor zoonotic diseases. And so I called her up on the phone and I said, Barbara, what do you think about this idea? Is it possible that antivirals that are used by humans go through wastewater treatment plants, get into aquatic insects, get into bats? Is that a possibility? Are the antivirals that we use to treat pandemics, are they a risk to the consumers of emerging aquatic insects? And Barbara got really excited, and Jurker. And what we did, this is kind of a paper without a lot of data. It's a lot of ideas, but we were able to take these ideas and calculations, taking stuff that we knew about to make some estimates. And I want to remind you that bats harbor beta coronaviruses, and so they may develop antiviral resistance. So this is a figure that Adrian made for our paper. Basically, there had been a study where they went and measured pharmaceutical concentrations all over the world. This great paper by Wilkinson et al. We overlaid those data points with where are bats, insectivorous bats. And what you can see is that where there are drugs in the water, there are definitely bats eating aquatic insects. There's no doubt about that. Like, that is the case. And depending on the bioaccumulation factor, what I mean by that is depending on how much the water concentration translates into how much concentration the bugs have, and then of course a whole bunch of other life history things, a bat, we did all this model, it's in the paper, that bats may consume more than a human dose of antivirals. This could lead, this is a total conjecture, it could lead to a spillover of an antiviral resistant virus that might be a pathogen to humans. We need to be careful. We only have so many antiviral drugs in our arsenal to fight them. If you remember back in 2020, before we had the vaccines, the thing that we relied on were antiviral drugs. We don't have very many antiviral drugs. Unlike antibiotics, where we have lots and lots, we have not very many antivirals. So we really need to be prudent with how we release them into the environment, in particular in the manufacture of them and in wastewater treatment plants. And now I want to tell you, lastly, a totally different kind of flux, because I think this paper is so cool. This was led by Megan Fork, who was a postdoc here, who's now in Westchester in Pennsylvania and is a faculty member there. And so I don't have that much time left, but I want to talk to you about what we did. In Baltimore, as part of the BES LTR, every week, like we do at Hubbard Brook, every week we go out and take water samples for chemistry. And we have this really beautiful record, long-term record. So one year I said, can we go out and measure drugs every week as well? So we did that. We took samples. My colleague Jurker Fick ran them in Sweden. And so here are the concentrations of drugs at one of the sites over time in the BES streams. We detected many different drugs. And in this case, they're delivered to Baltimore Harbor. So these drugs are getting into the streams through leaky infrastructure. There's no wastewater treatment plant in this basin. And what Megan did with some excellent chops in data analysis skills, data science skills, is she took the concentrations of drugs. She multiplied it by the discharge every time we took a sample. And from that, you can calculate flux. So it's similar to what I explained in the food web, but in this case, it's how much drugs are in the water. And what I'm just going to cut to the chase here. What we found is that in terms of expressed as equivalent human doses, it's every year, the leaking infrastructure contributes 35,000 doses of antidepressants to Baltimore Harbor, 30,000 doses of painkillers, and 1,000 doses of antibiotics. So when, yes, the concentrations are low, they're in the nanogram per liter, but when you think about it's like all the time, and you add it all up with how much water, it actually turns out to be a lot. The other thing that Megan did, which was like so smart that we came up with this approach that Megan did, is we routed these drugs through the infrastructure. So from human use, how do they get to the environment? What's the pathway? So this is a different kind of flux, but for, let's say, for acetaminophen, in the watershed we studied, there's given how many people and what we know about how commonly people take painkillers like acetaminophen, about 20,000 kilograms of acetaminophen are taken in by the human population that live in this watershed. Most of that acetaminophen, when you take acetaminophen, most of it is metabolized in our body, and not actually a lot comes out in our urine and our feces, but some does. A little comes out, and that little is actually kind of a lot of acetaminophen. Then most of that in this system in Baltimore, this where we found the leaks in the infrastructure is just that teeny thin line. Most of it goes to the wastewater treatment plant, which is another watershed over. And wastewater treatment plants do a great job of removing acetaminophen. So a lot of the acetaminophen is removed, but not all of it. And some of it comes out as leaks. So the leaking infrastructure contributes about 9 kilograms per year to the harbor. This is in contrast to the antibiotic trimethoprim. Trimethoprim, not very many people use it. It's not metabolized in our bodies. It goes to wastewater treatment plants, and wastewater treatment plants don't remove it at all. It goes right on through. And so the key insight for this in thinking about drugs and how they flux through systems is really important, because this has implications for management and river conservation. You want to not have drugs in your water, you got to fix the leaks in the infrastructure, and you have to upgrade your wastewater treatment plants. Because look at the load to the harbor is exactly the same, yet the pathway that they take from human use to the environment is totally different. So that's a key insight. So I'm going to close with some thoughts about what I've learned as an ecologist, as a person. I'm just going to start by saying that rivers are the best places to work. Rivers and streams are amazing. You can learn so much. They're just really cool, great systems to work in. I've had people ask me, what's your favorite river? And I say, whatever river I'm standing in is my favorite river, because I love them all, even from the Baltimore streams, which are, not that clean to, I love them all. They're great. Understanding fluxes is fundamental to understanding ecosystem processes, which I'm not the first one to say this. I'm just showing you things that I've done. So it's a really cool way of thinking about the way systems work. I didn't really talk about this, but this kind of work and ecosystem science in general does really benefit from contributions from all different disciplines. I think we benefit from all of our collaborations. We benefit from different ideas, and thinking about things from different perspectives really helps our science be better. I have really enjoyed working in large collaborative groups, from the BES group to Hubbard Brook to my teams in the Colorado River to the team in Kenya. It's productive, stimulating, and fun. I've only published one paper in my entire career by myself, and that was less fun than all the rest of the other papers. Like, it's just so much more fun to work with people. The data collection for fluxes is tedious. I'm not going to lie. Like, everything required, you got to I got to sample every week for a year. I got to sample all these things. But boy, it pays off in the end. You can really learn a lot if you take the time. Along the way, I also want to say a few other things. I've been fortunate to travel the world. I've gotten to work in, I guess every continent except for, I haven't really worked in Asia or Europe, but I've gotten to go there, and I haven't worked in Antarctica. But I've gotten to work everywhere else. It's been really great. And I've gotten to go to all these different types of rivers. And as people are thinking about their career and thinking about the arc of their career, I encourage people to go to different rivers. Working in the BES streams at the same time as working in the Mara in Kenya, it made me a better scientist. I thought better about the Mara and I did in Baltimore, because I worked in these totally different systems at the same time. You learn a lot. I've gotten to work with really great people. I also got to raise 2 amazing children, show you a picture of my beautiful children along the way, and I've had tons of fun. So What's next for me? Steve and I love to show this. This is like the World Atlas. We added the star. That's where we live. And this is the cover of the World Atlas. So if you think we moved to an interesting place, you're right, we did. That's where we live. It's beautiful. It's beautiful to live on a peninsula. Come visit us sometime if you find yourself in northern Michigan. That's where we are. And as you probably could tell from this, I'm shifting gears. I've become a painter, an artist, and it allows me to think about the world in a totally different way. To convey and portray nature through science requires measurement, whatever, and all these things that I did, which I so enjoy. But trying to paint a river and the flow of water and the movement is like a different way of engaging with nature. And I'm really excited to do that for the remainder of my life. If you want to look at my artwork, it's thanks to Steve Hamilton. He made me a website. You can see it all there. And with that, I'm happy to take any questions. And I want to thank everyone at the Cary for providing a wonderful place to work. And I've so enjoyed all of my years here. And then people online, all of my colleagues and students and everything have just made it just a real pleasure to have this as my career. So thank you so much. Thanks. Thank you. Thanks. Does anyone have any questions? And I'll repeat the questions. Yeah, thanks, Josh. Anyone have any questions? Or did I bombard you with too much flux? Hey, Chris. Oh, what flux haven't I measured that I would like to? Oh, that is such a great question. Now I'm going to give you the answer some later time in the future. Yeah. I mean, I don't know. I'll have to think about it, but that's a great question. Antivirals. Yes, we have not measured antivirals. I'm really hoping someone else will measure that. Yeah. Any other questions? Yeah, Kathy. Oh, thank you. Yeah. Yeah, so the question was, sorry, the previous question from Chris was, what flux haven't I measured? And I didn't give him an answer. So, but Kathy Weathers just asked me a question about, did we happen to measure mercury concentrations in the water and the sediments? We did not measure it in the water. It was challenging to measure mercury in the food web on a river trip without freezers already in the food web, but we were able to do it. But we didn't measure it in the water. Measuring mercury in the water takes a lot of there's this clean hands, dirty hands technique, which you, I'm sure, know about. So we did not measure that in the water. I don't believe we have it in the water, and we definitely didn't in the sediments, but that is a really great question. But I will provide like a slightly different answer to your question, which is, why is there mercury in the Grand Canyon, this national park? Like, why is it there? This like treasure? It's because of atmospheric deposition. So atmospheric deposition, mercury goes up into the atmosphere, it rains down. It's a very large watershed. There are coal-fired power plants in the region, and then just in general, you're hard-pressed to find any place in the world that doesn't have some amount of mercury in the food web. So, but yeah, that is a great question, Kathy. And yes, because probably the biggest flux of mercury, even if it's in a low concentration, is in all that water that's coming downstream, and potentially in the sediments as well. So yeah, that's a great question, but we didn't measure that. Yeah. Yeah, Evan. When you remove that from that trace supply, do you still find limitation, or how, I guess, what's the? Yeah. So right below the dam, so when we did the high flow experiment, it's not a flood, it's a high flow experiment. I should have said that properly. But when that was done, it like basically, I forget, we actually counted, we calculated how many New Zealand mud snails were actually exported. It's like in the billions, like because we measured in the water, we figured it out. But when we remove those, yes, and actually a bunch of other stuff got scoured away. We did get an increase in the production of prey of the black flies. But there's a lot of black flies because it's just rainbow trout eating them. And so when we removed the New Zealand mud snails, we did get an increase in trout production and in black fly production. And that's published in a paper by Cross et al. 2012 or something. And I'd have to go back and read our paper and figure out what we said. But it definitely it might not necessarily be the mechanism of just removing the New Zealand mud snail competition. It may just be like the scouring that occurred did increase the trout. In particular, the young of the year trout survived better. So if I recall, they were really, the increase in trout production was due to these small ones because there was more food for them. So yeah. Yeah.
Do you think that African rivers are fundamentally different than insect cycle because of the fact that Africa has kept close to this megafauna?
Oh, that's a great question. So I'll repeat the question, and I'm so sorry I forgot to repeat the question last time. It's hard. So the question was, do I think that the nutrient limitation in rivers, is that what you're asking me, in Africa is because of the megafauna that are still persistent in Africa? Well, I guess I didn't see your seminar last week, but I do want to point out that in the Mara we have hippos and we have wildebeest. But the hippo populations in Africa, they used to be all of sub-Saharan Africa. Their populations are really low compared to what they used to be in historic times. So their populations are extremely low relative to the way that they used to be. But I do think that Amanda led a paper, or Therese, we wrote a paper a couple of years ago called Legacies Lost and Found, and it's about this, in Kenya, we have these large wildlife input subsidies into the river, and certainly that is fundamentally different than rivers here. And we think of rivers as always having low nutrients in North America, but it may not have been that way when bison were roaming around. So yeah, I think that the role of large animals today is maybe, because they were gone before ecologists started measuring them. It's that shifting baseline problem, right? Where we don't know what the rivers were like 150 years ago while the bison were still there. So I mean, it would be great to be able to figure that out. But I do think that it's important to remember that certainly large animals in Africa and Kenya are restricted in range to what they used to be in the past, but they do remain. So sorry if I'm not really answering your question, but yeah. Other questions? Yeah, Denise in the back. How people go through septic systems in those communities, and is it different? Do you point it out or bust it out? Yeah, that's a great question. So I was asked whether or not, so we know that wastewater treatment plants don't entirely remove pharmaceuticals. That really sort of depends on the wastewater treatment technology that's in place there. But the question was, what about septic systems? And the answer is, I have not done that research, but lots of people are doing that kind of research. And there's some really cool work where people have like experimental septic systems. So like they build a septic system, on a university campus, and then they put stuff down the drain and they see what comes out the bottom. People have done surveys. It's definitely different. A septic system is going to be different in terms of what it removes and what comes out. But for sure, septic systems do not remove pharmaceuticals either. Like some of them might get removed well in a septic system, but they will release. The challenge with that kind of work is that septic systems are distributed, and it's very hard to kind of figure out in a watershed, like how many septic systems, what are they, how long have they been maintained? That's a really challenging thing versus here's the pipe, here's the wastewater treatment plant, I'm going to go take samples. So most of the people who have sampled pharmaceuticals in the water, they sample below wastewater treatment plants because that's a straightforward thing. So in Baltimore, we wanted to kind of address sort of a different issue, which is the leaks in the infrastructure. What about a system that has a bunch of leaks? What do the concentrations look like compared to a wastewater treatment plant? But that question of septic is, definitely septic systems. If you have a septic system You should not just put it in and think that it's going to work forever. You need to have it pumped. You have to make sure that it's still functioning properly. But even if it's functioning to its prime, the way it was designed, it still may not remove pharmaceuticals. That's a great question. Yeah, Sarah. Thanks. Oh, that's a great question. So Sarah asked me what was the most exciting part of my career and what was the most challenging. Let's see. The most exciting. I mean, I have long lists of exciting things that I've done, like daunting. When Bob Hall and I wrote a proposal to measure secondary production in the entire food web in the Colorado River, in a river we had never laid eyes on before, and then we went on a river trip in like big huge rafts with whitewater and everything, and it was like, what did we sign up for? Like, this is really hard. So like that was really daunting, and that was but exciting. I mean, I think for me the most exciting thing is when we have insight. I love all the exciting field work, that's great, but especially when I'm working with a postdoc or student and it's like, we could do this, we could make this calculation, we could figure this thing out, like Megan figuring out like, oh, we could route this all the way through the system and see what's happening, or Amanda, Chris, like we could figure this out. Like that to me is the most exciting moment when you're working with your colleagues and you say, if we just did this, if we took this measurement, we could learn something totally new. To me, that's the most exciting. Challenging, the biggest challenge I have to say is funding work on pharmaceuticals and personal care products. Hands down, that's the hardest part. I'm really proud of all that work. Super challenging to fund it. There's not really a great place to go get money to do that kind of work. So a lot of it was done, as we could, but I'm really proud of all that work. We accomplished a great deal, but there wasn't really ever a great funding mechanism for that. So that was pretty, because if you want to do science, you have to have resources to pay people and get out in the field and take samples. So, but that's a great question. Thanks, Sarah. We're over time, so I don't want to keep people from eating lunch. Shannon, are there any questions from online?
There is one from AJ Reisinger, who said that when he was working with you one time, you said that do it all again, you go back and do geology.
Oh, thanks, AJ.
Well,
actually, yeah,
he'd like to know how you feel now.
Okay. No, I think what I told AJ, and Ariel gave me a book when she left, Ariel Sjogren, who is now at University of Alabama. I worked with her as well. What I think I said is, if I could do another PhD, I would do it in geology, and I think it's because all that time going down the Grand Canyon, Colden Baxter was my collaborator. We brought him in because we realized we really needed his expertise. And I joked that like the first trip was geology 101. Then it was 102, 103. I kept asking him questions and learning about geology, and I really love geology. And but I love what I do. It just also I don't really know much about geology, but boy, rocks are cool and change over time. And you could study flux of rocks, so that would be pretty neat. Yeah. Thanks, AJ. Thanks. Thank you all. Thanks for people joining online.